A pleasant day ahead, everyone. Today, we will be discussing with you about the patch clamp technique. So, the patch clamp technique is an essential tool in electrophysiology for studying ion channel behavior in various cell types, including neurons, muscle fibers, cardiomyocytes, and oocytes. It enables the evaluation of single ion channel conductance by forming a high resistance seal with the cellular membrane and removing a patch of a cell membrane containing the ion channel of interest. The technique also involves the application of current and voltage that will be discussed by the next reporter. The process typically includes the preparation of solutions, cells or brain slices, as well as the pulling and polishing of the recording electrode. The application of patch clamp technique requires a specialized equipment and software for signal acquisition, amplification, and digitization, depending on the research interest. Different configurations of the patch clamp technique can be utilized, such as the cell-attached patch clamp recording and general principle of patch clamp recording, which we will also discuss later on. The patch clamp technique has also been combined with live cell imaging approaches and is widely used to study ion channel activity and conductance. It has become one of the most important tools for understanding behavior of single cells or whole cellular networks in the nervous system. This technique has roots in the pioneering work of prominent scientists like Luigi Galvani, Peter Moeller, Hermann von Helmholtz, Alan Lloyd Hodgkin, and Andrew Huxley.
Good day everyone. So today I will be discussing the cell-attached patch clamp recording. So cell-attached recording in which a patch electrode is attached to the cell but the membrane is not broken. So cell-attached patch clamp recording, when the pipette is in the closest proximity to the cell membrane, mild suction is applied to gain a tight seal between the pipette and the membrane. So in order to form the cell-attached mode, a pipette tip is placed on the surface of the cell, so forming a low resistance contact with its membrane. So, slight suction applied to the upper end of the pipette result in the formation of a tight seal, such as a seal with resistance in the range of gigaohms is called the giga seal. So, in the cell attachment, recordings are made from the membrane area under the pipette, while the structure of the cell remains intact. So, a metal electrode inside the glass pipette containing the salt solution resembling the fluid normally found in the cell. So, accomplishes transduction with the ionic current into the electrical current, while another one in the bath solution serves as a ground point, and AG to AGCI electrodes are specially used for the low junction potentials and weak polarization. So, the tight seal between the pipette and the cell membrane isolates the membrane patch electrically. So, which means that all ions fluxing the membrane patch flow into the pipette and are recorded by an electrode connected to the highly insensitive electronic amplifier. So, a bath electrode is used to set the zero level.
So what is general principle patch clamp recording? The general principle of patch clamp recording refers to the underlying concept and methodology of a technique used in electrophysiology to study the electrical activity of individual cells, specifically the behavior of ion channels in the cell membrane. Patch clamp recording allows to measure and analyze the flow of ions through these channels, providing insights into the function of cells and their physiological processes. The technique involves using a glass pipette which is filled with an electrolyte solution to form a tight cells with a small patch of the cell membrane. This cell allows for the isolation and measurement of electrical currents generated by ion channels within the patch membrane. By applying different modes of recording such as cell attach or whole cell configuration, it can investigate various aspects of ion channels behavior. So this is a patch clamp recording with a focus on the role of the glass pipette. And here how it works. First one is the pipette preparations. The process begins with the preparation of a glass pipette, and the pipette is pulled from a glass capillary tube to create a very fine tip, usually less than a micron in diameter. This pipette is then filled with an electrolyte solution that mimics the cell's internal environment. The second one is the cell attachment. The pipette is brought into the contact with the cell membrane using a micromanipulator, a device that allows for very precise movement. The fine tip of the pipette is pressed against the cell membrane and a small amount of suction is applied. This forms a tight cells between the pipette and the cell membrane known as a gigacell. The formation of this cell is crucial for the success of the experiment. Next is the mode selection. Depending on the research questions, different modes of recording can be in cell attach mode. The patch of membrane inside the pipette remains intact, allowing the study of ion channels in their natural environment. Next is the whole cell mode. More suction is applied to rupture the membrane patch, allowing the study of ions flow across the entire cell membrane. Next is the other modes like inside out and outside out. It allows the study of ion of one side of the membrane in isolation. The fourth one is the recording. Once the desired mode is achieved, the ion flow through the channels can be recorded. The ion flow creates tiny electrical currents which are detected by the pipette, amplified, and then recorded. The glass pipette serves as an interface between the cell and the recording equipment, allowing the measurement of these tiny currents. The fifth one is the data analysis. The recorded data is then analyzed to understand the properties of the ion channels, such as their conductance, selectivity, and the factors that influence their opening and closing. So that is how it works. So, the role of the glass pipette is a crucial part of the patch clamp technique, serving as the interface between the cell and the recording equipment. It allows for the isolation of a small patch of the cell membrane and the measurement of ion flow through that patch.
So hello, I am Riza Logroño. So my assigned topic for today is all about the current clamp. So current clamp is one technique of patch clamp. So what is current clamp? So current clamp is the fixed transmembrane voltage change resulting from ion channel activity. So this technique allows the investigator to control the amount of current injected into the cell, so thereby controlling the transmembrane potential. So in current clamp mode, we act like tiny current conductors, injecting precise electrical currents into the cell through the patch pipette. So this injected current mimics the natural currents that occur during various cellular activities like neuron firing or muscle contraction. So by monitoring the resulting changes in the cell's membrane voltage, we can observe how the cell responds to different current stimuli. So imagine poking a cell with a tiny needle that also pumps in current. So the cell, surprised by the influx of ions, free back with its own voltage fluctuations, which we can then record and analyze. So this is like listening to the cell's electrical conversations in response to other currents routes.
Voltage clamp. It is used to study substrate selectivity of ion transporters by measuring ion currents. To conduct a voltage clamp experiment, a portion of the axon, which would include the cell membrane and all the voltage-rated ion channels located there, is removed from a neuron and placed into a solution that mimics that of physiological extracellular solution. The initial step in the voltage clamp method is to measure the membrane potential of the axon. A recording electrode is placed into the axon, and a reference of electrode is placed into the extracellular solution. The voltage difference between these two electrodes is the membrane potential of the axon. The researchers running the experiment can set a desired membrane potential for the cell. The equipment then compares the desired membrane potential with the measured membrane potential from the electrodes. If these values differ, current is injected into the cell to change the measured membrane potential and make it equal to desired potential. The equipment continues the cycle for the length of the experiment. It constantly measures and compares the actual membrane potential with the desired potential, and then uses current to correct any changes, clamping the potential of one value.